Good afternoon, everyone. On behalf of the Hepatitis B Foundation and our program, Hepatitis Delta Connect, I want to thank you for joining today's webinar. My name is Sierra Boder, and I am the Program Manager of the Hepatitis Delta Connect program here at the Hepatitis B Foundation. We're excited to present today's session, Hepatitis Delta Treatment Endpoints. How do we measure success in the era of emerging therapies? One second. Before we get started, I'd like to review a few housekeeping items and let you know how you can participate in today's webinar. Please note that all attendees are in listen-only mode and will be muted during the presentations. There is also a phone-in option, and the call-in numbers are on your screen. Please note, you may also submit your questions in writing during the presentation by typing them in the question box on your GoToWebinar control panel. We will be reviewing them as they come in, and we will be holding a Q&A period after the presentation. And finally, today's webinar is being recorded, and you will receive an email with a link to view a recording of today's presentation after the webinar. Now I'd like to introduce our presenter today, Dr. Ohad Etzion, who is the Director at the Department of Gastroenterology and Liver Diseases at the Soroka University Medical Center in Beersheba, Israel, and he'll be presenting on hepatitis D treatment endpoints. How do we measure success in the era of emerging therapies? And with that, I'd like to turn it over to Dr. Etzion.
Thank you for the kind introduction, Sierra. I would also like to thank the Hepatitis B Foundation for inviting me to speak about this important topic. I don't seem to be in control of the mouse.
Are you able to advance it?
No, not yet.
How about now?
Let's see. No, not yet. Okay. Maybe you should start advancing the slide if there seems to be some technical issue.
Yeah. Okay, Dr. Etzion, would you mind if you just said next slide and I can go ahead and advance it for you?
Okay, so let's start.
Sorry about that.
Oh, it's okay. Okay, so please, next slide, please. Can you advance the slides?
Oh. Yes. Okay, there we go.
Okay, so next slide. Okay, so the outline of this talk will go through several points. First is the epidemiology and some important clinical aspects of hepatitis delta infection. Then we move on to discussing current management and its limitations. The next topic will be defining suitable endpoints for clinical trials in hepatitis delta virus infection, and finally we'll briefly review data from recently completed studies and the outline of the design of the upcoming trials evaluating novel therapies for hepatitis delta. Next slide, please. So hepatitis delta is an incomplete virus that is completely dependent on the presence of the hepatitis B virus envelope proteins to package its genome and produce infectious particles. And next, please. So this paradigm has recently been challenged by an interesting paper published in Nature Communications earlier in 2019, showing that the envelope viruses distinct from hepatitis B virus, such as the Hep C virus and other viruses, can propagate delta infection in the liver of co-infected humanized mice for several months. Whether this bears any clinical significance in humans is yet to be determined, but it raises the question, or the thought, whether we should test for hepatitis delta in clinical settings of hepatitis of unknown origin. Go back, please. Okay.
Sorry about
back. Yeah, it's no. You're moving. I think you're moving forwards instead of backward. Okay. All right. Next slide. Thank you. Okay, there are around 15 to 20 million people affected by Delta worldwide. It is estimated that around 5% of the global HBV infected population, or around 240 million people, is also co-infected with Delta, which amounts to around, as we said, 15 to 20 million people worldwide. But the distribution of HBV prevalence is not equal in all regions, with pockets of very high prevalence identified in regions such as Africa, the Amazon basin, Eastern and Mediterranean Europe, Middle East, and parts of Asia, such as Mongolia, where around 60% of the HBV population is co-infected with the virus. There are 8 different HDV genotypes that have been identified, with genotype 1 being the most commonly distributed genotype worldwide. Genotype 2 and 4 are seen mainly in the Far East. 5 to 8 are considered the African genotypes, and 3 is seen in Northern and South America. Now, HDV is generally found in any country that we test for it, and if we don't find it, then it was either not tested for, and we definitely know that the rates of HDV screening is poor in many countries, or that the platform for testing, the kits, are not valid. Next slide, please. So in the early 90s, there was a notion that HDV prevalence is declining, which was attributed to the implementation of HBV universal immunization, a program in many countries. However, recent waves of immigration to Europe from outside of Europe and inside Europe to Central Europe, and also in other Western countries, show that there is higher prevalence described in recent years. And next slide, please. So it's therefore not surprising that in most countries in Central and Southern Europe, the majority of infected individuals with Delta are actually immigrants from countries with a higher HDV prevalence. It is also estimated that the true prevalence in these countries is probably higher because part of the immigrant population arrives illegally in illegal ways, and consequently do not have access to high quality medical services which provide testing for hepatitis delta. Next slide, please. Now, HDV is the most severe form of chronic viral hepatitis. While the evolution of chronic viral hepatitis follows a common pathway starting with liver and neck inflammation, which progresses through increasing stages of fibrosis to cirrhosis and end-stage liver disease with its complication, the portion of patients following this course and the pace at which this sequence of events occurs is quite variable between viruses. So with Hep C, it occurs in 10 to 20 or maybe 30% of patients over a course of 2 to 3 decades. In Hep B, the timeline is shorter, 5 to 10 years, in 20%, but in Delta, the rate of progression to cirrhosis is fast, and it occurs in 2 to 3 times higher than in other viruses such as Hep B and C. Next, please. Next slide. This is somewhat provocative. Go back again. Yeah, so it's a somewhat provocative slide illustrating the impact of HDV on survival. 2 of the 3 survival curves presented are of 2 types of malignancies, and the third one is an HDV survival curve over time. And as we can see, the survival curve of Delta patients resembles more those of that of patients with advanced cancer than of patients with chronic infectious diseases. And this fact seems to be under-acknowledged even among very experienced clinicians. Next, please. So the cumulative incidence of both the development of cirrhosis and liver-related events, such as bleeding varices and SVP in Delta patients, is higher compared to HBV monoinfected patients. And also in the lower panel, we can see that in the EuroHep viral hepatitis study showed that after adjustment for all potential prognostic variables by Cox model, the estimated 5-year probability for development of HCC in HBV patients was more than 3 to 6 times higher than HBV monoinfected patients, depending on their e antigen status. Next, please. Next slide, thank you. So another indication for the increased severity of chronic hepatitis delta compared to B monoinfection is the proportion of liver transplants performed for each of these indications. The data presented here is from a single transplant center in Israel, in Jerusalem, showing that the number and rates of liver transplantation for each indication in Israel, each indication, B versus D. And in Israel, the background prevalence of Delta seropositivity is around 6.5%. And the data obtained from the center clearly shows that there is an overrepresentation of Delta-infected patients in the liver transplanted population compared to the background prevalence in the general population. This also suggests, again, that progression rates to advanced liver disease are higher in this population. Next slide, please. So the survival rates following liver transplantation seem to have to be better in Delta than compared to HBV monoinfected and HCV patients. And this has been attributed in the past to low pre-transplant levels of HBV replication in Delta versus B patients, and probably leading to a lower likelihood of a HBs antigen reappearance in the transplant. Well, unfortunately, we all know that while this data is encouraging, liver transplantation is not a viable option for the majority of HDV-infected patients with advanced liver disease, so we need to find other solutions. Next slide, please. Okay, so let's move on to discuss current management of hepatitis delta. So unfortunately, since its discovery in 1989 by Risetto and colleagues until this very day, there is no treatment approved for Delta by FDA or any other regulatory agencies. And treatment with interferon alpha has been tested in multiple studies assessing its efficacy, the efficacy of standard interferon earlier, and then with pegylated interferon. And these have been conducted over the years with variable results. The two largest randomized controlled trials evaluating pegylated interferon alpha in chronic hepatitis delta are the HIT 1 and 2. And next, please. So in the HIT 1, it was a 3-arm study of pegylated interferon alpha with a Defovir or placebo, and then a Defovir only arm administered for 48 weeks with a 24-week follow-up, with the primary endpoint being the combined achievement of undetectable HDV RNA and ALT normalization at end of treatment. This endpoint was achieved in only 7% of patients in the interferon arms and none in the Defovir arm. HDV negativity at week 48 was achieved in around a quarter of patients in the interferon arms and in none in the Defovir arm. And in the HIT 2, the treatment was extended to 96 weeks and was combined with either tenofovir or placebo. And the primary endpoint was end of treatment HDV RNA negativity. And it was achieved in 48% and in the combination arm, interferon and tenofovir, and in only 33% of interferon placebo. So extension of treatment showed little increase in HDV RNA response, and the mean HDV RNA levels almost plateaued during the second year of therapy. And so we can see that treatment is not very efficacious, also involves a lot of side effects, and is limited to patients with compensated liver disease. And let's see what happens after treatment is stopped. Next slide, please. So the question arises whether a sustained virological response akin to what we see in hepatitis C is feasible with interferon alpha. And in both HEATED 1 and HEATED 2, 6 months follow-up showed that 28 and 27% response in terms of HDV negativity, which is pretty modest, but even more discouraging was the fact that in a longer follow-up of several years, more than 50% of patients that were HDV negative at week 24 post-treatment became HDV RNA positive at some time point during that follow-up. So we understand that the concept of sustained virological response is probably cannot be applied in this disease in the same manner as we do it in hepatitis C. Next slide, please. Okay, so before, sorry, okay, but despite the fact that the virus is not eradicated in the majority of patients, several studies show that interferon alpha is associated with improved long-term clinical outcomes. The study published in 2017 showed that interferon on a long follow-up, several years, patients treated with interferon did better in terms of survival compared to patients with no therapy or patients treated with nukes. And in a Greek study, patients followed for 13 years showed that the hazard ratio for liver-related events in interferon alpha treated patients versus nuke therapy and non-treatment was 0.14%, which was statistically significant. Next slide, please. So before diving into the discussion on clinical endpoints for HDV studies, let's take a step back and clarify the difference between goals of therapy and endpoints, which is not quite the same. And now, physicians, clinicians who treat patients with chronic liver disease, our focus is often shifts to focusing on lab indices such as ALT and viral load. And so we tend to forget that the overarching goals of therapy are not to eradicate the virus infecting the liver, but rather to prevent disease progression and the development of end-stage liver complications and death. And these don't always parallel. For instance, we tend not to treat HBV patients that have high viral load with persistently normal liver enzymes, those that we used to call immune tolerant patients, because the disease does not seem to progress in this setting. The problem is that it takes many years to measure events that capture these ultimate goals of treatment. It's something that isn't within the scope of pivotal studies leading to drug approval. And that's the reason why we need to use surrogate markers that are reasonably likely to predict clinical benefit on the long run. Next slide, please. So such endpoints have been very well established in chronic hepatitis C and hepatitis B. In HCV, sustained virological response has been shown to have a strong association with improved survival. And in Hep B, the ultimate surrogate endpoint predicting long-term clinical benefit is, of course, achievement of S antigen loss, the equivalent of functional cure. But this is seldom achieved with current therapies for HBV. But we know that persistent on treatment viral suppression, although a less favorable endpoint, has nevertheless been shown to be strongly associated with improved clinical outcomes, such as reduction of HCC and fewer end-stage liver disease complications, such as varicose bleeding and SVP. Next slide, please. Now, furthermore, what we've learned from studies on HBV therapies in the last decade is that a prolonged on-treatment viral suppression, even without viral eradication, is associated with the fibrosis regression, even in cirrhotic patients. So continued treatment, even without viral eradication, can be beneficial, at least in hepatitis B. Next slide, please. So what about hepatitis delta? So we need to take into account several facts and concepts. They should be acknowledged before discussing selection of endpoints in clinical trials. First is the data on specific surrogate endpoints that are associated with long-term clinical benefit is pretty sparse. and so we may need to make some assumptions derived from data obtained from other liver diseases, especially hepatitis B monoinfection. The second point is that we are not yet sure whether cure from HDV is feasible, and if so, what are the endpoints that clearly define viral eradication in HDV? We saw earlier that a sustained virological response, determining sustained virological response 24 weeks after stopping therapy is very problematic. The third concept is acknowledging the difference between ideal and reasonable endpoints in HDV therapy. Because of HDV dependency on S antigen, then loss of S will most likely lead to HDV eradication and cure. But this, at the current stage of drug development for HDV, S loss may not be accomplished in the foreseeable future, at least in most drugs tested now. And therefore, we need to select primary endpoints defining successful therapy that are more reasonable, but still associated with presumed clinical benefit. Next slide, please. So what are the components we believe should be captured as endpoints in upcoming trials for HDV? These include measures of virus suppression, such as viral load decline and or virus undetectability, markers of improvement in necroinflammation, such as ALT normalization, and if patients biopsies, we can see improvement in histology scores. The composite endpoints, such as combining viral suppression with necroinflammation, are probably better than each of these indices alone, because there's probably a higher likelihood that the combination of the two will be a better predictive of clinical improvement. The last concept is that durability of response, which is the impact of any therapy following its cessation, should also be measured either as primary or secondary endpoint, because it is reasonable to assume that long-lasting effect of any drug will probably have a better effect on clinical outcomes. Next slide, please. So recently, a group of experts in the field has joined efforts and published a white paper, a position paper, in an attempt to outline reasonable and realistic recommendations for selection of endpoints for clinical trials in Delta. And the consensus of this group reached was that the 2 or more log reduction in HDV viral load at the end of treatment compared to baseline should probably be the target for assessment of initial treatment efficacy with the new drugs that are currently being evaluated. This recommendation, next please. Okay, so this recommendation is largely based on a study published by Farsi and colleagues showing that patients treated with interferon alpha who had a 2 log reduction or more in HDV viral load had a better long-term outcomes, and we basically relied on that paper for this recommendation. Next, please. And the expert panel also defined several virological, biochemical, and serological treatment goals to be measured as secondary endpoints in clinical trials. And these include on and off treatment viral suppression and decline, ALT normalization, and improvement in liver inflammatory scores, and also a serological marker of S antigen levels decline or disappearance in S antigen seroconversion. Next, please. They also suggested to capture several exploratory endpoints, such as liver stiffness, intrahepatic HDV RNA, and immune markers that may give additional evidence on the effect of new therapeutic interventions in Delta. Next, please. Now, regarding measurement of histologic improvement, we know that it happens in hepatitis B. There is very few studies showing such an endpoint can be achieved in Delta infection, at least with interferon alpha. One of those was a small study comparing standard interferon to lamivudine, showing that compared to lamivudine therapy alone, the majority of patients treated with interferon showed histological improvement, or at least no worsening in histological scores at end of therapy. Another evidence came from the HDV trial mentioned earlier, which showed that prolonged therapy with Pegylated interferon, 96 weeks, led to regression of fibrosis. But measuring histological improvement in clinical trials may be problematic for several reasons. including the patient's compliance with multiple biopsies and some technical issues, such as obtaining sufficient tissues for analysis, which is very common in HDV population, in which many patients are already cirrhotic at baseline. So the quality of the biopsies may be pretty low. And this may affect data gathering and patient dropout from these studies. So while histological improvement may be an important endpoint, that will be determined in the future, predicting that could predict future clinical benefit, it should probably be a secondary rather than a primary endpoint in clinical trials of Delta infection. Next slide, please. Next slide. Thank you. Okay. The FDA has also recently released a draft guidance for the industry outlining concepts for consideration when choosing efficacy endpoints for clinical trials in hepatitis delta, and the FDA recognizes that at the current state of knowledge, we need to rely on endpoints that are reasonably likely to predict clinical benefit. We just don't know enough. And they define preferable endpoints as those that combine undetectable HDV RNA with ALT normalization, that composite endpoint we described earlier, but also acknowledge that a 2-log reduction in HDV RNA plus ALT normalization is an acceptable endpoint of efficacy. In addition, the FDA acknowledges that the optimal time point for primary endpoint assessment is currently unknown, whether it's end of treatment or a certain time frame after stopping treatment. And it's separated between therapies that are intended to be administered indefinitely versus those that are given for a finite duration in terms of on and off treatment assessment of efficacy. Next slide, please. Okay, so now let's move on to discuss the novel therapies that are currently being developed for Delta hepatitis. And as mentioned earlier, one of the major barriers for development of effective therapies for this disease is the fact that unlike hepatitis B and C, Delta has no RNA polymers to target. So new treatments need to target other steps in the virus's life cycle. And so currently there are 4 drugs for HDV that are being evaluated at different stages of clinical development. These include Merculex and B, an inhibitor of viral entry, Lonafarnib that interferes with viral assembly, a nucleic acid polymer, the REP compound that interferes with HBs antigen release, and a new immunomodulator, the Pegylated Interferon alpha. Next slide, please. So let's start with Merculex B. This is a first-in-class entry inhibitor for treatment of chronic B and D infection. It's composed of a synthetic 47 amino acid and acetylated 3S1 lipopeptide that targets the sodium taurocholate co-transporter polypeptide, the NTCP receptor, which is the bile acid receptor of the cell, the hepatocyte. It also serves as the receptor to which enveloped hepatitis B and Delta viruses bind to before being internalized into hepatocyte. And following engagement, the drug blocks receptor function of NTCP and subsequently prevents or assumed to prevent the HBV and HDV virus entry. Next, please. Okay, so Merculex B was evaluated in a number of phase 2 trials. First, in a pilot study of 24 patients, the drug was administered subcutaneously. That is the way it's administered in a lead-in period of 24 weeks, either alone or in combination with Pegylated interferon alpha, and with another arm of interferon alpha monotherapy. The primary endpoint was S antigen decrease at 12 weeks of therapy. This endpoint was not met. However, there was a meaningful drops were seen at week 24 in the Merculex B arms, with the most pronounced one was in the combination arm of Merculex B and the interferon monotherapy. Next slide, please. The next study was an open label phase 2 study of 120 patients dosed in 4 arms, assessing that was a dose finding study of 2, 5, and 10 milligrams of Merculex B combined with tenofovir for 24 weeks, and then nuke-only arm as a comparator. The primary endpoint was a 2-log decline in HDV RNA or RNA negativity at end of treatment. All 3 intervention arms showed viral load reduction at the end of treatment, with the most pronounced effect seen in the 10 milligram arm. In addition, ALT normalized in 40 to 50% of patients, but there was no change in S antigen levels, and the virus level increased during follow-up period, and it was concluded that probably longer periods of treatment are needed to achieve viral clearance. The drug was tolerable in most patients. There was an increase in bile acid, but without pruritus. Next, please. Next slide. Thank you. The MIR-203 phase 2 study looked at 2 doses of Merculex B that were combined with Pegylated interferon, and another arm received 2 milligrams of Merculex monotherapy. And there was also an interferon monotherapy arm. All received treatment for 48 weeks. So in this study, treatment was extended. The primary endpoint was more ambitious, undetectable HDV RNA 24 weeks post-treatment. They wanted to measure durability of response. This endpoint was met in a considerable proportion of patients in the 2 combo arms, but in none of the 2 milligram monotherapy arm. In that arm, the viral load was seen on decline was seen on treatment, but most patients returned to baseline values on follow-up. In addition, there was an ALT decline seen in all intervention arm on and off treatment. So the conclusion from the study was that the combo therapy is more efficacious than monotherapy and may even might induce cure in a subset of patients. Next slide, please. The MIR-203 had an extension study, the results of which were presented at the last AASLD meeting. In this study, additional arms were explored, 10 milligrams once daily dose, combined with Pegylated interferon, and a 10 milligram monotherapy administered twice daily as a 5 milligram each. In the left lower panel, just a minute. Okay, yeah. In the left here, it's actually Pegylated interferon monotherapy and bulevirtide. This is the name of the commercial name of the drug. Bulevirtide 10 milligram. And in the right lower panel, 10 milligrams of bulevirtide plus tenofovir is compared to 2 milligrams monotherapy from the original study. And as seen, the percentage of HDV negativity reached at the end of treatment is high, 87% in the combo versus 40% in the monotherapy. And interestingly, ALT normalization was more pronounced in the bulevirtide monotherapy arm compared to the combo arm. Now, results of end of follow-up are still awaited, and it would be very interesting to see whether these impressive numbers are indeed durable. Next slide, please. So based on the accumulated data obtained from these phase 2 trials with Merculex, the Merculex company has recently launched a phase 3 registration trial, recruiting 150 patients in sites in the US, Russia, Germany, Italy, and Sweden. And the study design includes 3 arms of Merculex combined with a nuke, with the first arms starting with an observational period of 48 weeks. Followed by 96 weeks of therapy that are then followed by another 48-week follow-up period. This is a very long study, and one of the reasons for that is that mathematical modeling and previous studies expected that the virus might be eliminated with longer duration of therapy. Now, the other 2 arms are composed of 2 doses of Merculex, 2 milligrams and 10 milligrams, combined with a nuke, administered for 2 years. And the primary endpoint is a composite one of undetectable HDV RNA or a 2-log decline in viral load, plus ALT normalization at week 48 of therapy. Next slide, please. Next compound we will discuss is Lonafarnib. Lonafarnib is a prenylation inhibitor, and prenylation is a lipid modification that involves addition of a prenyl lipid to proteins, which result in promotion of membrane association and protein-protein interaction. And in a landmark science paper by Jeffrey Glenn, published a decade or two ago, it was shown that the interaction of large hepatitis delta antigen with HBs antigen involves prenylation of the large delta antigen, and that Lonafarnib disrupts this process, which subsequently prevents formation of secreted HDV virus particle. Now, in a proof of concept study conducted at the NIH, 14 patients were dosed for 28 days with Lonafarnib 100 milligrams or 200 milligrams versus placebo, and it was shown that a significant HDV RNA decline occurred, but with considerable GI side effects in the higher doses. But there was no evidence of virological resistance, and that makes sense because the drug targets the host protein and not the viral protein. And also, it encourages us because that means that if there's no resistance to the drug, it could probably be employed in repeated cycles of treatment or a salvage therapy. Next slide, please. Next slide. Thank you. This proof of concept study was followed by a series of 4 phase 2 trials for identifying the optimal dose and regimen for future registration of this drug. Next slide, please. The LOWR HDV1 study was designed to assess the tolerability and viral response of different doses of Lonafarnib as either monotherapy or in combination with ritonavir or Pegylated interferon. Now, Lonafarnib is metabolized by the CYP3A4 enzyme, and ritonavir is a CYP3A4 inhibitor. So combining the two drugs allows administration of a lower drug doses of Lonafarnib that still reach high circulating drug levels, while at the same time there's less exposure of the drug to the GI tract, which improves the drug tolerability. Its main side effects are diarrhea and weight loss, and so this is the purpose of this combo. And the primary endpoint of the study was HDV RNA decline between baseline at end of treatment, which was at either at 8 or 12 weeks. There were multiple groups tested with different doses and combinations. And the results showed that the combo therapies, either with ritonavir or with Pegylated interferon, showed the most prominent viral decline in ALT normalization, and there was improved tolerance with GI tolerance with the lower doses. Now, there was viral rebound in all but 2 patients who had ALT flares, which were followed by HDV becoming undetected and then sort of flying under the radar, either being undetected or at the lower limit of quantification for a longer period of time. And this might suggest that the immune system may take part in viral clearance in a subset of patients treated with this drug. Next, please. Now, the LOWR HDV was a dose-finding study exploring high and low doses of Lonafarnib combined with ritonavir with or without interferon. And treatment was extended to 24 weeks, and the aim was to find the optimal dose of combo therapy with ritonavir as all oral regimen or with Pegylated interferon as a combination therapy in terms of efficacy and tolerability. The primary endpoint was a HDV RNA decline at end of treatment. Now, results showed that the 50 milligram all oral dose was superior to 25 milligram all oral, and that at 25 milligram dose of Lonafarnib, the combined effect of on viral decline of interferon and the 25 milligram dose was better than 25 milligram all oral. Additionally, the most synergistic effect was seen in the combination therapy of Lonafarnib, 50 milligrams with ritonavir and Pegylated interferon. All oral therapy seems like an attractive option in patients with low baseline viral load to begin with. We can see in the left lower table, the table that shows that in patients with low viral load, those with below 4 logs at baseline, all oral therapy could achieve viral levels below limit of quantification or above 2 logs in all patients, 100% of patients. In patients that had high viral load to begin with, more than 4 logs, they responded very well to combination therapy with Lonafarnib, ritonavir, and Pegylated interferon. 50% of them were BLQ below limit of quantification, and 88% achieved a 2 log or more decline at week 24. Next slide, please. Two additional small studies, the LOWR 3 and LOWR 4, were explored all oral regimens as either single dose or dose escalation manner. In LOWR 3, there were 3 placebo arms followed by higher and lower doses of Lonafarnib plus ritonavir. The primary endpoint was declined at end of treatment, and it was most prominent in the 50 milligram groups. This is the repeating theme. You can see that the 50 milligram dose has an advantage. Additionally, in a subset of patients with a triphasic kinetic response, mathematical modeling predicted that treatment beyond 6 months may lead to viral clearance. Now, the GI side effects were generally mild in the lower doses group combined with ritonavir. In the LOWR 4, the dose escalation study, dose was escalated from 50 to 75 to 100 milligrams in a stepwise manner, and patients, as long as they could tolerate and maintain these doses for 24 weeks. Response rates of more than 2 log decline in HDV RNA were seen in 27 and 20% of patients at week 24 and 48, respectively. And ALT normalization was seen in over 50% of patients at the end of treatment, but all patients had ALT was again elevated. Also, 53 of patients with the higher doses needed dose reduction during the study, and 13% had to discontinue because of GI-related side effects. So the higher doses again were less tolerable. In the lower doses, there were GI side effects such as diarrhea, weight loss, but these were mild, and patients did not discontinue therapy because of that. Next slide, please. Now, based on the accumulated data from the LOWR program, Eiger Pharmaceuticals, this is the company developing this drug, has recently launched a phase 3 4-arm study, recruiting 400 patients in over 100 sites in US, Europe, Middle East, and Asia. This is by far the largest HDV clinical trial ever conducted. This study includes an all-oral arm with 50 milligram, the most optimal dose found in terms of efficacy and tolerability in phase 2 studies. Another arm of a combo therapy of Lonafarnib, ritonavir, and Pegylated Interferon, and 2 additional arms of Interferon monotherapy and a placebo arm. Now, all treatments are administered for 48 weeks, followed by a 24-week follow-up. And here too, the primary endpoint is a composite one, combining 2 or more log reduction in HDV RNA with ALT normalization at end of treatment. Now, additionally, patients will undergo paired liver biopsy in the study at baseline at the end of treatment, and histological improvement will serve as a secondary endpoint in the study. Next, please. Okay, next compound is Pegylated interferon lambda. This is a novel first-in-class type 3 interferon that binds to a unique receptor that is different from the type 1 interferon, like interferon alpha, which is highly expressed on hepatocyte and very limited expression on hematopoietic and CNS cells. It uses a similar downstream signaling pathway as type 1 interferon, and it's actually been developed originally by BMS for treatment of hepatitis B and C before the advent of DAAs, and it was tested in more than 3,000 patients in several clinical trials, showing comparable antiviral activity with less of the typical side effects seen in interferon alpha, such as bone marrow suppression, depression, and autoimmune phenomena. Next slide, please. Now, the LIMT phase 2 Lambda monotherapy study was a 2-arm study. Patients were randomized, but it was an open-label study. The treatment was administered in 2 doses, 120 micrograms and 180 micrograms once weekly for 48 weeks, followed by a 24-week follow-up period, with the objective being to evaluate the safety and tolerability of Lambda monotherapy for 48 weeks. The efficacy endpoint was a change in HDV RNA from baseline to week 48 and week 72. And as we can see in the left lower panel, there was a meaningful decline in HDV RNA at end of treatment, minus 2.3 logs, and it was more pronounced in the 180 microgram group. Additionally, what was very encouraging was that the post-treatment week 24, meaning week 72 of the study, 36% of patients showed levels below limit of quantification, and 36 showed an ALT normalization. Next slide, please. And the durable virological response, the week 72 time point, was even results was even better, reaching close around 50% in patients with low viral load baseline. This was seen in subgroup analyses. Again, the patients with viral loads below 4 logs at baseline showed a more favorable response. In terms of side effects, this was something it's very important because we compare it to historical data of interferon alpha. So in terms of side effects, the vast majority of grade 1 side effects were grade 1 or 2 and were associated with constitutional symptoms, with only one incident of neutropenia in a single patient. No bone marrow suppression of any sort seen in other patients. None of the patients received growth factors like we used to treat patients treated with pegylated interferon alpha. Now, there were a couple of incidences of hyperbilirubinemia in 9 patients in the study, mostly from patients coming from Pakistan. And this required dose reduction or terminating the drug, but without any clinical consequences such as hepatic decompensation. And all patients that stopped therapy, bilirubin declined and came back to normal levels pretty quickly. And in an analysis, it was suggested that a transporter-based mechanism is probably responsible for these bilirubin elevations in genetically predisposed individuals. Now, we did a follow-up kinetic analysis of viral kinetic in patients treated in the study. And the viral kinetics found some unique patterns seen in Lambda versus alpha from historical data. Interestingly, in patients with biphasic HDV RNA response, the HDV RNA decline was not accompanied by S antigen decline. And this is opposed to kinetic data from interferon-treated patients with a biphasic response, in which there is a decline in HDV RNA paralleled by a decline in HBs antigen levels. Now, concluding this, the results of this study is that the durable virologic response was seen in 36% of patients, and this compares favorably both in terms of efficacy to interferon and in terms of side effect profile. Now, we didn't biopsy the patients in the study, but recently 2 of the patients participated in the study in our site were biopsied in order to enter the D-LIVR study, and surprisingly, these 2 patients had a 2 and 3 point reduction in fibrosis score compared to their historical biopsies performed more than a decade ago. Now, if this represents a true phenomenon, it is a very encouraging sign that Lambda may influence fibrosis regression, at least in a subset of treated patients. Next slide, please. Now, these encouraging results triggered the LIFT study. This study was a phase 2A open-label study combining Lambda with Lonafarnib. Lambda is also developed by Eiger, so these 2 compounds were combined in the phase 2 study. Treatment was administered for 24 weeks, and the primary endpoint was 2 or more log reduction in HDV RNA at week 24. Okay, and looking at the results, we can see that therapy with Lambda, Lonafarnib, and ritonavir was relatively safe in most patients. Okay, and per protocol, there were discontinuations of triple combination therapy, but mostly due to side effects of related to pegylated interferon alpha. And now, in terms of efficacy, the results showed that the response rate was 53% BLQ, and 95% had more than 2 log decline at end of treatment, suggested that combining these 2 drugs has a synergistic effect on the hepatitis delta life cycle, and it's definitely worthwhile pursuing this drug combination in future studies. Next slide, please. Well, the last compound we'll review is the REP-2139. This is a nucleic acid polymer. Nucleic acid polymer, or NAPs, are oligonucleotides with a broad spectrum of antiviral activity that is not sequence specific. NAPs are proposed to bind to amphipathic protein structures. And in hepatitis delta, we don't quite understand what is the exact mechanism of action, but it is assumed that, or suggested that these particles bind to both S antigen and allows host-mediated clearance of S antigen. Also, it may bind to the large and small delta antigen with potential upstream inhibition of HDV virus synthesis and assembly. Next slide, please. Next slide. Thank you. Replicor, the company developing this drug, is developing it for both hepatitis B and delta. And thus far, a single clinical trial was conducted in 12 patients with hepatitis delta. And this study involved a treatment with the lead-in REP-2139 for 15 weeks, followed by addition of pegylated interferon for another 15 weeks as a combo therapy, and then another 18 weeks of interferon monotherapy. And as noted during the first phase of the study, in the REP-2139 monotherapy, there was a rapid decline in S antigen level. This observation is indeed unique to this drug, as opposed to all other pipeline drugs currently being evaluated. In 3.5-year follow-up, S antigen levels below limit of quantification were maintained in 42% of patients, and 36% of patients showed HBs antigen seroconversion. HDV RNA response of more than 2 logs compared to baseline was maintained in 82% of patients, and in 2/3, RNA was undetectable at the end of follow-up, again, 3.5 years. These are encouraging results, and they definitely call for further development of this compound. We have to take into account that this is a very small study. And indeed, next slide, please. Replicor is currently planning to launch a 4-year phase 2 study in Q4 of 2020 that will explore both IV and subcutaneous treatment with Replicor 2139, combined with magnesium, which is a more tolerable compound than the current Replicor molecule that is binding to calcium. These will be combined with tenofovir, with or without interferon, and it's a long study, a 48-week treatment phase, and then another 4 years follow-up. So results will be waited for in a couple of years. Next slide, please. Okay, so after this somewhat exhaustive review of current therapies, it's time to conclude our talk. And in summary, we can say that chronic hepatitis delta is a severe disease for which current management is unsatisfactory. Data on surrogate endpoints predicting long-term clinical benefit is sparse, but we are definitely gathering new data as the ongoing trials are progressing. And now, until more data accumulates, clinical trials assessing novel therapies for delta will need to rely on endpoints that are reasonably likely to predict clinical benefit. And long-term follow-up will be required to establish the validity of these endpoints as surrogate markers of future clinical benefit, meaning survival and a decrease in liver-related events. Now, finally, there is definitely a room for hope with these new therapies on the horizon. Next slide, please. And this is probably an opportunity. I'd like to take this opportunity to thank all the dedicated investigators worldwide, as well as our partners in the industry and the regulatory agencies that are all joining forces to advance research and hopefully promote a cure for this important disease in the future. I also would like to thank the patients participating in past and current trials and their families for their support, and also encourage patients with Delta infection to enroll to one of the ongoing and future clinical trials conducted across US, Europe, Asia, and other countries, because the faster we complete enrollment and completion of these studies, the sooner we will have these drugs available for routine clinical use. Thank you.
Thank you so much, Dr. Etzion. That was a fantastic presentation. We'll now begin the Q&A portion of our webinar. As mentioned at the beginning, we will start by answering questions submitted to us via the chat box. If you would like to submit a question now, please do so using the question field on your screen. We did have a few questions come in during the webinar, and we'll jump into the first one. If nukes don't affect hepatitis delta, why are they often used in clinical trial treatment protocols?
Well, this is a great question, and the main reason is that we know that delta suppresses hepatitis B. So if we treat delta and the treatment shows efficacy, then when delta is suppressed, HBV might reactivate. And while these flares may be beneficial in some patients, they may be dangerous in other patients. So at least in clinical trials, from a safety standpoint, we have to suppress hepatitis B in order to prevent these events that might be detrimental to patients.
Thank you. Our next question is, can we monitor the response to treatment with ALT measurement? We do know that NASH can increase ALT as well. So is there a role in measuring ALT in treatment other than a decline in the RNA?
Yeah, well, actually, this issue was debated among the experts writing up the white paper. And yes, indeed, in patients with delta, as other patients and other patients in living in Western or Westernized countries, have a high prevalence of fatty liver in 30–40% of the population. So the question arises whether if ALT is increased, whether treatment of delta can really capture, if ALT is a surrogate marker, can it really capture response that is associated with delta, or if we had another liver disease that might affect ALT, that wouldn't be a good surrogate marker. So if we, at least in the two phase 3 studies, we use a composite endpoint of ALT normalization plus HDV RNA. So these two biomarkers are linked to one another. So there's a higher likelihood that if both of them are declined following HDV treatment, that it's more associated with HDV than with another liver disease such as fatty liver. Now, ALT is a very strong marker of improvement across all studies in liver disease, whether it's viral hepatitis, autoimmune hepatitis, or other diseases. So we still believe that as a marker of biochemical response, it should be included, and the FDA is on board with us.
Thank you, Dr. Etzion. In the interest of time, we'll just ask one more question. Is there any hope for an interferon-free or all-oral therapy, or will one drug ever do the job, or will it always be a combination with interferon?
Well, as presented earlier in one of the Lonafarnib trials, there was a subset of patients that benefited from an all-oral therapy, those that had a low viral load to begin with, and of course having an all-oral option and not without a combination with interferon is something we desire, because even treatment with Lambda is limited to patients that have compensated liver disease. So hopefully, in the phase 3 study of Lonafarnib, we'll see improvement or efficacy reached in patients that are treated in the all-oral arm. Also, the other combination, other drugs that are currently administered as subcutaneous, like Pegylated or the NAPs that are IV or will be subcutaneous, may be developed as oral formulations in the future. And also there might be other oral drugs coming in the pipeline. time will tell. But we definitely hope that this eventually, in the long run, will be a viable option for treatment.
Great. Thank you so much, Dr. Etzion. And I do apologize to the audience. Since we're at the top of the hour, we'll conclude the Q&A portion. But please, if you do have another question, please don't hesitate to contact us directly. We want to thank everyone for participating in today's webinar. And we do encourage you to fill out the survey. It also includes opportunities for patients and providers to receive additional information about clinical trials and free testing for hepatitis delta. So please look out for a message from us after, which will also pop up with additional information. Again, thank you. We greatly appreciate your participation and feedback as you exit the session today. Thank you all again, and have a great rest of your day. And thank you, Dr. Etzion, for your time and expertise.
Thank you, and thank you. I would like to thank the audience for participating and asking questions.
Thank you so much. Have a good day, everyone.
Mm-hmm.